Thank you, Sate, and welcome all of you to this first session of the day, which is about disease control in wild populations. And we will hear about killing the animals, depopulation, but also non-invasive methods. The first speaker is Jørn Våge. He's actually a colleague of mine in the Norwegian Veterinary Institute in Oslo, where he is a wildlife veterinarian. And he is in charge of the surveillance program against chronic wasting disease in Norway. Jørn has the two first presentations today, but he will merge them into one. And he is free to take any pause if he wants between them. So, welcome.
Thank you very much, Cecilia, and the organizers for inviting me to this seminar. I think it will be very interesting. Welfare in wild animal is, of course, has always been there, but it has not been addressed that much. So I think it's on time, and I hope it will be good. As Cecilia said, I will talk about chronic wasting disease in Norway, and I will also address a little bit more general for the culling, eradication of wild animals for disease eradication, and try to at least, I will not give that much answers, but I will try to lift questions around the welfare issues in such situations. But I've been working with chronic wasting disease since we detected that in Norway. This is the grim phase of chronic wasting disease. I will give a short background for the disease understanding because it's not everyone is familiar with chronic wasting disease and this disease group being a prion disease. But this is elk, Cervus elaphus from North America. They are kind of the same as ours, but a little bit bigger, our red deer. So it's the red deer brother in the North American continent, which is sick from chronic wasting disease in a quite late stage of the disease. Chronic wasting disease is a prion disease in cervids. It's a transmissible spongiform encephalopathy. It's a chronic neurodegenerative disease, and it's completely lethal. And it's special in the case that, I can say we have, this is chronic wasting disease is a disease in cervids, but we have the counterpart in sheep, which is scrapie, and also in the bovine, which is BSE, and also in humans with Creutzfeldt-Jakob disease. And in humans, we also sadly acquired the BSE type in humans with the variant Creutzfeldt-Jakob appearing after a big outbreak of BSE in the United Kingdom in the late 80s. Very few people, but still there is a couple of hundred people that has died from this variant Creutzfeldt-Jakob disease, which is kind of special. So, but that's for the transmissible spongiform encephalopathies. BSE is the only one that has been proven to be to transfer to humans. Otherwise, scrapie and chronic wasting disease is still understood as not a zoonotic disease, but it's an animal disease. It's special in the way that prion diseases, it's talking about proteins. We do not have any agent that is like a bacteria, virus, or fungi carrying any nucleic acids, meaning DNA or RNA, to be transmitted. But it's a host-encoded cellular protein called the prion protein, which changed conformation into a protein that is more stable, meaning that the body is not able, the enzymes in the body is not able to degrade this protein, and we will have an accumulation. And that is happening. You need the cellular protein to be transformed. So it's not in the way that it's reproducing this misfolded protein. It's not reproducing unless you have presence of the cellular protein. And it will be able to accumulate in different parts of the body, the cellular prion protein it's abundant, especially in cell membranes. The cellular function it's a little bit blurry, but it's present there. But the problem arises when it accumulates in the central nervous system. Like here on the right, there is a brain from the very first reindeer that we detected in Norway, showing the red spots here. That's the visualization of misfolded prion proteins in an immunohistochemistry staining. Special with the case of chronic wasting disease, at least what we now define as the classical chronic wasting disease. I will show you that we have discovered something after the detection of this disease in reindeer in Norway. We have started to test and we find something. But the classical type of chronic wasting disease, also for classical scrapie in sheep, is special in the form that they have a very long incubation time, where in the period where the animals appear healthy, but they do shed prions, the infectious agent in this disease, through saliva, urine, and feces. And it's shed into the environment, and also it's shared with their relatives. So you will have a long, more or less healthy incubation period. You will enter a disease state, and you will have a shorter period of an animal showing more and more severe symptoms. They will die. There is no cure. There is no possibility for any vaccination. It's special with the prion diseases that they do not evoke any immunological reaction in the body. So it's, in that aspect, difficult. And also, the enzymes are not able to cope with this accumulation of proteins. They die. And a further adding of the complicated system is that these prions, they will be infectious from the environment for a long period of, we don't know exactly for how long, and it will probably, most probably, depend also on the different environments and so on, but there will be a contamination of the environment for years. I will give you a short background from the North American continent about this disease, because it appeared in Colorado. That was in a captive animal. Of course, we did not know at that time that it was a prion disease. That has been defined in the 80s. But in 67, they started to describe a syndrome that they called chronic wasting disease in deer. It developed, and around 2000, this started to spread. And it also emerged in wildlife, so it spilled over from the captive animals till wildlife. And of course, captive animals and transport of captive animals have been a contributor to the spread of the disease in USA and Canada. It was exported to South Korea with elk from Canada around 2000. And the last 20 years, there's been a huge development in the disease spreading in the North American continent. And today, they face 26 states in the US and 3 provinces in Canada where they have detected this disease. So it's just spreading. And that's the problem also when it has become endemic. It's very difficult. I don't believe it's possible to get rid of this disease in the North American continent. In Norway, we have had for a long time surveillance of also chronic wasting disease, the prion diseases, because after the outbreak in Great Britain with the BSE in bovine, there was, of course, the awareness of the disease transmitting to humans, and prion diseases was for sure put on the map, and there's been put a lot of effort into to try to protect humans from having prions entering their food chain. And we have had a surveillance program since the beginning of 2000. And through this surveillance program, there was this detection with the disease in Norway in 2016. This was the first appearance in Europe, and it was all outside North America, apart from South Korea, where they imported diseased animals. And it was also the first appearance in reindeer. It has been experimentally transferred to reindeer, but not in the wild. So this was a very special situation. Of course, with the background from the US and Canada and the understanding of the disease spreading and the severeness of the disease, of course, there was a huge amount of awareness in Norway, and we tried to explore what this could be. There's been a lot of scientific going through the knowledge about chronic wasting disease and also the other transmissible spongiform encephalopathies in bovine and sheep, and trying to understand what could this be and what would the hazards and consequences be. So there has been a lot of support for the management from the scientific community in Norway. This finding led to a decision of a political decision based, of course, on the science, quite uniform advice from both veterinary and biological communities, that we had to take away this herd which we have detected. It was a herd of wild reindeer in a quite confined area, mountains, a small alpine mountain area. So we decided to take out the whole of this subpopulation. It was about 10% of the total wild reindeer population in Norway. Why did we give this advice? Of course, because of the history of the North American continent, the understanding of the severeness and this cycle that I showed you with spreading of the disease, accumulation of environmental contamination, and the possibility that this could transfer to all different cervids. So, and our understanding was that we were in a beginning of this outbreak. We had never seen this before, and we have quite Norway is, as Sweden and Finland, a nation of hunters. We are very much outside, outdoor, and we work with the cervids, we observe them. We had never seen anything like this. So we were quite sure, and also from calculations, based on knowledge from the North America, we estimated that we would have maybe in this herd 1% prevalence. So, meaning that we were in the beginning of an outbreak, and we tried to take it seriously. Also, the fact that reindeer is I could almost say hypersocial species. They clump together. If this would be the reindeer, this could be the moose. If you take away half the reindeer, they will still go together and transmit the disease. If you take away half the moose, they will be spread and you will have less transfer. So that was one of the prerequisites for having the possibility to succeed. You had to take away all of them. And also because of this environmental contamination, of course. And being a serious disease which is listed, of course the authorities could not just let it go. We have a lot of cervids in Norway, in Scandinavia and the Nordic countries as a whole. It's about 120,000 moose. We have a lot of roe deer, we have a lot of red deer. We harvest from them, 30,000 approximately from each species each year. Additionally, we have this wild reindeer, which are managed in these 23, it's now one more, 24 different areas which are green. Those are management areas for wild reindeer. So it's in the southern part of Norway. Nordfjella with the culled population is this area in the central of this. Additionally, we have from the mid of Norway and northwards, we have a lot of production of semi-domesticated reindeer. Of course, there is a lot, and this will extend into Sweden and Finland, being in Lapland and the Sami culture, which is crossing the borders. This production, we have about 250,000. The number is not always correct, but it's in that size of semi-domesticated reindeer, which of course was also vulnerable for this disease. We have, as I said, we have had a surveillance program from 2000 till 15. Our records show that we had tested just about 2,000 animals. Of course, in 2016, with the emergence of the disease, we increased this, and a lot of effort was put in, and now, this was just the other day, we tested, so we have passed 92,000 samples since 2016 in the surveillance program to try to get the knowledge of the spread of the disease. We have cases in moose, we have cases in reindeer, and one red deer. The reindeer is here, the red deer is there, and the moose is scattered a little bit. These are Swedish moose, this is a Finnish moose, or Finland tend to say it was a Russian moose, but it was found in Finland. So these are all, as far as we know, similar to the Norwegian moose. And as you start looking, you will also find, and that's interesting, because scrapie, in 98, it was discovered that scrapie had also the classical scrapie in sheep that had been there for several hundred years with symptoms. But we also detected, when we started to test and we started to look at this, what we have, we also define atypical scrapie, which is different from the classical form, in the meaning that it's not transmitting horizontally, at least not detectable in a detectable way. So we, in 2018, we published a paper telling that what we have found in the moose was different. It's a prion disease, it's transmissible, but it's different in the appearance in the animal, and we called it atypical a bit because it looked like it was more similar to atypical scrapie. And then we started to call the chronic wasting disease seen previously, also in the reindeer, we started to call that classical, just to differ between these two types. So it's different strains, it's a typing. It's of course, if we make an analog to a virus, you have a lot of different subtypes, and you classify them. We also do that with prion strains. And in the reindeer, we have, all of them are in, well, they are between 1.5 years up till 8 years, those 19 animals that we detected during this culling situation. In moose and red deer, it's all females only, and they do not have any distribution of these prions into the lymphatic tissues. So it's, as far as we are able to detect with the normal detection methods today that we use in diagnostics, they are within the central nervous system. For the reindeer, and also what has been seen in North America, there is a distribution throughout to the lymphoreticular system, and this is, we also hold this as a important character in the spreading and transmission horizontally of the disease, because if it's in the lymphatic tissue, it will also be able to be shed and spread. So, sorry, these are just different characteristics of the two different strains. Moose is moose, hopefully, and reindeer is now red deer. It's not moose, but it's more similar to moose than it is to the reindeer. So there are differences. And we try to, when we try to strain type prions, we have certain biochemistry measures that we do. There are weights of the protein, how they transfer in a gel. There are different pictures, but the true typing and the full, or till now, the full understanding of typing is that we make use of bioassays. This is a bank wall. I don't know if you have this in Sweden and Finland, but it's for children. This is very well known klatremus. And bank walls, they are naturally susceptible to prion diseases. As I said, you need the cellular protein to be converted. Bank walls, cellular protein will be converted for a lot of different prion strains. For mice, we genetically engineer them so that they express the protein of the species that you want to study. For example, cervid or humanized mice. So they express human cellular prion protein or cervid prion protein. So, and then when we inoculate these models, they will eventually develop disease. The typing will then be for the incubation time, for the lesion profile, and so on. And we do this as successive inoculations to have a stable line. And when we have stabilized these lines, we can say that this line of prions, this strain of prions, they appear in this manner in this model. And when we test them, we can say, okay, this belongs to this strain. So that's the typing of prions. Interestingly, we had in 2018 also the emergence of prion disease in camelids. That is the first description. It's from North Africa. And of course, camelids is a very important group of animals around the world as working animals and also for food. So that was scary, and it's few animals, but they are described to have distribution to the lymphatic system. So we don't know actually, but this was the first description of prion disease in camelids. So you can question whether TSE, the transmissible spongiform encephalopathies, are these in animal an emerging group of diseases? So I think we have had scrapie in sheep for 200-300 years described in symptoms. We had the appearance of chronic wasting disease in 1967 in the US. Now it emerged in Europe. We have had it in humans for as long as we know in different forms. So I think that chronic wasting disease and the prion diseases, that's a picture and a chapter of a book that is just, we have just begun to write that chapter. Yeah, I will not spend that much time on this. It's just that we also try to, because the Norwegian Veterinary Institute we are an institute that supports the management of disease in Norway. The ministry and also the Food Safety Authority in Norway rely very much on our knowledge and support in the scientific advice on how to handle diseases. And we try to drag this over to the animal welfare aspect. We are working with our epidemiologists to try to model and to infer how this disease will develop, and also try to model how many animals you have to test, meaning kill, because it's a post-mortem diagnostics. You use the brain tissue, so you have to kill the animal to get the knowledge about contamination. And we try to model how many do we have to test to obtain a certain level of disease freedom, or a security for disease or infection freedom. So that's important in animal welfare aspect. There is, for chronic wasting disease, it challenged the Norwegian system a bit, because we haven't had really large outbreaks. Of course, we have the avian influenza coming in, which was managed, and that's a wild species. We have had history of fighting diseases in fish, which are wild animals. But this was maybe the first big encounter of a disease that had to be bridged between different governmental bodies, because the health regulation is for the Agriculture Ministry and the Food Safety Authority, and the species, the wild species, they are managed, and the regulation is managed by the environmental authorities. So they had to work together, because this has been a huge operation, testing these 90,000 animals. It's quite a work. So they had to cooperate in a new way, which I think was good, and I think it has made us more robust to face different diseases in the future. The Food Safety Authority, they have, together with the environmental side, they have, of course, they had to make a plan for the eradication of these 2,500 animals, plus minus, that was in Nordfjella, the infected population, to take that out. But there was also very clear, because this is a defined area, for wild reindeer, there should be a replacement of these animals. We have said that it has to be at least 5 years from the last animal going out till we replace, because we don't know about this, for how long this environmental contamination will be there. Okay, so that was for the chronic wasting disease. I will swap to the second part of the talk, eradication of wild animals. As I said, we don't have very many examples of that practice in Norway. We have had, for sure, we have been working with salmon, try to getting rid of Gyrodactylus salaris. It's killing the small salmons. It's a organism that will affect the gills. And we have been using rotenone, which is, and you take out whole rivers, and they kill everything that breeds with gills. So it's the fishes and all the small organisms that use gills. So you eradicate not only the fish, but also all the other fauna with gills. That has been questionable in many aspects. We have been working with that for decades. They now say that they have succeeded, and we have rivers that we declare free of the gyro. But of course, it's a welfare aspect in the way that you kill a lot more than what you just want to get rid of. The reindeer, this was the gyro. The eradication plan. Of course, when we were saying that we are going to take away 2,500 wild reindeer, you have to do that in a way. We should do it with regular hunting in the beginning. We should follow up with hunters, special hunters organized from the authority. But we also had a discussion whether it would be feasible to try to collect these wild animals into rounds and having them fenced and slaughter them to be able to do this in a short time, because we had only one winter that it was decided that they should be away. We should be able to eradicate them in one winter. So it was discussed, and that could be very welfare, in a welfare aspect, difficult to say that you are going to collect several thousand wild animals into herds and fence them and slaughter them. It would be quite a chaos. We were lucky that we didn't have to go there, because we managed to kill them one by one through professional hunting. This was transparent in the way that we had a website put up where we had a log. So the professional hunters from the government, every day they were writing up a lot of different things, how many shots they've been used to kill how many animals, how many kilometers they had been driving with the snowmobiles, the weather conditions, and a whole lot of different parameters that they were logging to show that they were transparent. Because of course there was discussions, there was opposition towards doing this at all, and it was of course also opposition in how this was supposed to be done. And in the midwinter, the animals are living in an alpine area, it's cold, it's scarce with food, and they are already in a stress situation. They will be pushed to their limits, and to assure that there was some kind of surveillance of their welfare. The Food Safety Authority also had dedicated people following this process and checking and writing reports during the whole process, releasing these reports to the public on the website to let them know that we try to do this as transparent and as good as possible. So I think that was very important to try to, because it was a lot of criticism, of course, that the government is just going in, they do what they want, they use helicopter, they use snowmobile, they stress these animals. Of course it was a stressful situation, but we wanted it to be, we didn't see any options, so then we wanted it to be transparent. So this was on the website of the Food Safety Authority, saying that they will take the animal welfare and surveillance this in Nordfjella in the action of the culling. Another example is we don't have very much wild boar in Norway. I think we have about 1,000. Sweden have at least 300,000, and we have sharing a long border. And we are now establishing it's not called an eradication plan, but it's a management plan against wild boar. Meaning, and the saying is that we are going to have as few wild boars on the smallest spot possible in Norway. So this plan is just being, I think it was deadline yesterday or tomorrow, yeah. So it's on its way, but in that aspect, when you start to establish such a plan, the animal welfare is of course very important, because hunting and everything will be, depending on your goals, you might be a little bit labil in the way that you accept things that would maybe not be the best for the animals. This is the last picture I have. It's from a mox-osk calf. We have a small population of muskox in the central south of Norway, in Dovrefjell. I was there and we were going to take samples from animals, and in that aspect there is a plan, management plan for these muskox. They are to be held at around 200 animals. So we use this plan and the number of the population size to cull animals, and we have to take it down. So it's a population regulation. But we do also then use this population regulation to take out diseased animals. So in a way, we are fighting disease, but it's not merely just because we want to fight that disease, but it's also for population regulation purposes. But this is ORF virus, scurv, ectyma, there are different names. It's a zoonosis. And of course, you can imagine when you are going to eat small, short grass in an icy coverage, it's not very easy to eat with this phase. So, and it was not, it was very bad condition. So we shot this animal. It could not follow his mother. So, but this is the salmon, the chronic wasting disease, and in the future the wild boar, and also these mox-oxen are situations where we use culling as a means of taking out also for disease. So you can question, is it good to be killed as a wild animal? It depends on the state you are in, maybe. Yeah. Do we have time for questions, or was I?
No, we have actually 6 minutes for questions, and I'm sure there are many. Lotta?
Yeah, Lotta Berg, Swedish University of Agricultural Sciences. I have a question about the reindeer herd that was infected. You said that they were all killed by hunting eventually, but that you had a plan B for using sort of more of a slaughter method. If you had ended up in a situation where you would have had to apply that, would you have actually sort of stunned them and slaughtered them the standard way, or would you have done it differently because of saving the brain for sampling? Because otherwise you shoot them with a captive bolt or a rifle in the head, and that would make sampling more difficult.
That was one of the difficult questions that we had to answer, because definitely we didn't want to shoot them in the head or bolt them. So we had the discussion whether it was feasible, how we should kill them, avoiding destroying the brain for sampling. So And I don't know if we actually landed that question completely, but I think we were to, in a way, stab and...
I think we ended up by using captive bolt.
Okay, so that was the end, yeah. And then when you sample for chronic wasting disease or TSE in animals in general, you take a spoon sample, you go into the foramen magnum, the big hole, you cut the head off and you go in, and you have a spoon and you collect from the brain stem. So
which you may still be able to do after captive bolt stunning.
Yeah,
possibly.
It's, yeah, trying to sample from a headshot animal during hunt is difficult, but it could be possible, yeah.
Margareta.
My name is Margareta Sten, and I'm from the Swedish Center for Animal Welfare, but I'm also a mood scientist. It's very interesting to hear you, as all times I've been listening to you, and I will listen to you in a couple of weeks too. But when we are in Finland, it's really interesting, about what you think about chronic wasting disease and the forest reindeer herd that's very dangerous, and also white-tailed deer that they have here. It is really challenging, I think.
Yeah, of course it's challenging, and luckily Finland has not detected anything but that one moose, which was an old female, showing no distribution to the lymphatic system. So the situation for Finland is as far as now, they don't have detected any classical chronic wasting disease. So, but of course, you have a lot of, we have in the Nordic countries, we have a lot of cervids, and to our knowledge, all of these cervids are susceptible to both types. We have no scientific meaning of that they are not susceptible to either one of them. So it's, I think it's a very severe situation for the whole of the Nordic countries, definitely.
Then it's one in the back.
Hello, my name is Francisco from the Wolf Action Group. I would like to ask you if you have studied predation as a method of disease control by large predators in Norway.
It's, I wouldn't come to Norway to do that because we don't have very much large carnivores. So we are a country of sheep, so we have few large carnivores, and in this area where we have had chronic wasting disease, you don't have large carnivores for predation. So we have not been able in Norway to study that thoroughly. Of course, on the border to Sweden, we have some wolves, we have some few bears, but to use them in a mechanism of disease control, we don't have that kind of populations of carnivores.
Okay, I have a little comment. I'm Sanna Sainma. I'm from Helsinki Zoo. I'm veterinarian. I'm working also with the forest reindeer introduction program. So I really understand your question, because forest reindeer is an endangered species. But the good thing that maybe prevents that a little bit, because you couldn't take you can't take the whole culling of the whole herd as a method, but then you lose the species. So what we do in Finland, we actually have a fence between the reindeer herding area, partly between the reindeer herding area and the areas where we have forest reindeers. And then we do the culling if there is forest reindeer that goes to the reindeer herding area, because we are allowed to have reindeers only in the northern part of Finland, but then south from that area we have forest reindeers. So we try to keep these two populations apart, so that's maybe a little protective factor in that.
I think it would be very difficult, because we are now with the Nordfjella, it's the northern part of, it's a divided area, and the southern part of that, which is divided by a road. We have a smaller population of about plus minus 500 animals, which we are now testing thoroughly. But they are tested after hunting, But then we try to, by the epidemiologist, we try to model how many we have to test to ensure freedom of infection. And we try to manage that way. Of course, if these animals in such close vicinity to the infected population or the previous infected population, if we would find anything there, I would say as a scientist in this manner, I would say it would be advisable to take away that, but we also test bigger areas farther south, and then the picture would be completely different. So it depends on the, of course, the size and how valuable that population would be. But the fact is that this kind of disease is much more challenging than any disease I know that is infectious. So it's with the environmental contamination and the persistence of the disease, and that it's you have no means by trying to get rid of it unless you take away the infectious material, meaning you have to remove those animals. So it will challenge our understanding of how to manage diseases in wildlife. In sheep, then it's a less problem. You cull them and you clean the area. But in the wildlife, it's difficult. But this is more than challenging.
Yeah, and there is
something
going on with forest reindeer as well.
We have to move on. So we'll give Jørn an applause again.